Okay.
Okay, so our last speaker is David Holcman from Normale. So right now he's in the UK for an extended visit, I guess. He's going to tell us about electrodiffusion and dendritic spine.
Okay, thanks, Sam. First of all, I would like to thank all of you to be here for the last talk. I'm actually replacing somebody that could not come. It's not only the last talk of the last day of the last workshop of the program, it's the end of the program actually. And it was a great pleasure to organize this, to have all of you here, to have all the people that have accepted to come here for the Rothschild lecture, for the extended period, or not only just being here for the workshop, but for months of the program. This was a great, really something great for us, and I hope that this will foster not only collaboration, but create a community which is difficult to emerge when we looked at the classical mathematics, classical PDEs, and suddenly we are saying, no guys, there is now a new life for PDE. It's not anymore existence and uniqueness, but asymptotics. So this is one thing. And also, the revolution in the past 10 years in data analysis cannot be ignored. This is certainly a revolution. But as Asaf showed you, I think the two are very important to understand really how the biology is working. We are competing with people that have microscope, that can do genetics, and still we believe that we can see features in the data that without the mathematics you cannot see. So we still have to push, and I'm very happy that we are all here and trying to move into that direction. Okay, that's enough for all of this. I would like to discuss electrodiffusion in dendritic spine, having both a theoretical approach to understand something that I still do not understand how it works. It's about how charges, ions really evolve from nanometer to micrometer. And I would like to dedicate all of this to my long-term collaborator, Zev, who has been sick for a year, and I hope everything will be okay. So let's discuss, let me give you the framework for all of this. We're discussing here neuronal cell, neuronal communications. A neuron, as you know, is made of basically three parts: the dendrites, the soma, and axon that deliver the information, some of the information to the next neuron. We still do not understand. I mean, we means the entire community. It's still an important challenge to understand how do they work, especially with the problem of aging. So a particular question is about memory that we had a very nice lecture, I think, on Wednesday about what is memory. So I would like to redescribe a bit this. So this has to do with a little structure here that you are seeing, which are called dendritic spines, which is the place where two synapses are connecting. So before that, let me remind you the discovery Nobel Prize of the 80s of Neher and Zachman. You can stick an electrode inside the soma, and what you can see, you can record. And you look at the electrical activity. This is a neuron embedded here in a network. And you can see basically that there is some activity. The voltage, the membrane potential, can basically here go between two states, up and down state, which is affected by drugs, as we know, and it can be irreversibly be affected. And I would like you to pay attention about the time scale. This is for this guide of the order of 10 milliseconds. So basically, electrical activity occur at a time scale of 1 to 10 milliseconds. This is much faster than diffusion. And yet, we still have, it's not clear what are the processes, because obviously, ions are involved. Diffusion of ion are involved in generating such a signal. So let's now focus on this particular interaction between two neurons, presynaptic terminal, postsynaptic terminal. So upon some action potential, here you have the signal which is going to be transduced from electrical activity to chemical activity by the release here of a neurotransmitter, about 3,000, that are going to move by diffusion. And there was in the last 5-10 years some evidences that diffusion is not necessarily enough. You can have also here the effect of the electric field that is pushing the, for example, if we think about an excitatory connection, glutamate, which is excitatory neurotransmitter, to some specific spot here, which are called receptors. And when they are bound to the receptor, they open, and then there is a flux of ion in this postsynaptic terminal here in yellow. So, what is memory? This is, again, a very complex question. So, in the last 30 years, especially in this country and in the United States, there were a development about the mechanism. And they took this place, the changes in this place, as reflecting learning and memory, and this is called synaptic plasticity. And basically, the protocol that you have already been shown 2 days ago is that if you stimulate a cell, here's a specific neuron, and you record to another neuron, and they are just connected by those synapses, then if you send a stimulation here and you record here, what you see here is such a current which reflects the interaction between the pre and the post. This is a signal generated in the post terminal by the release of neurotransmitter that I have just described. Now, under some specific condition protocol, you can, for the same stimulation, after you do this protocol, you generate a current which is much bigger, and this is stable over time. And you can reverse this. That is, you can go from this to this stage by a different protocol, and this is called LTD. Now, it took probably 10 years to understand what happened between here and here at a molecular level. And this was a change in the number of receptors at the synapse. That is, here you have, let's say, 5 receptors, 5 umpire receptors. Sorry. Now I cannot go back. Yes. So you may have here few receptors, let's say 5, and now, due to this mechanism, you have 10. It's still the fundamental question here: how do they arrive here? Why they can anchor and stay stable at these places while being moving all the time by Brownian motion? So there are conceptual questions that are still to be solved. So the question I would like to ask today is that, is the receptor increase sufficient to explain the change in this postsynaptic response? That is it just a molecular effect, just we have added new receptor and that's it? Or something else is happening, because ultimately we are talking about electricity, and electricity that is going to be recorded in the soma, that is far away from this little location. So for this, let's try to add now some kind of electrical component which is very different from electrical circuits, where the electricity is carried by moving ions. Now, if you open Wikipedia or any classical textbook, Bertil Ille, the books that has been written for the last 20, 30, 40 years, you get this concentration of potassium, sodium, which are the main 2 players. Calcium is very, it's an important player, but not in terms of the concentration. And then you have chloride. So this is what happened. You have inside the cell, and this is usually the concentration in the extracellular space. Do we have electron neutrality here? If you add the charges, there is no compensation. So, what's going to be the description if we want to understand what is the voltage, what is the current generated in this microdomain? How are we going to, what is the right model, first of all, to start with, to generate a system where you don't have electron neutrality? And then you can even ask the question, at which level this is important? Are we talking about nanometer, tens of nanometer, hundreds of nanometers? And just to give you a flavor of how things happen, after you have an action potential, sodium goes in, potassium goes out, but the potassium is not coming back to neurons. It is first absorbed by glial cells, and then there is a cycle. And the time scale of an action potential is usually much faster than the recycling of all of these ions. And in some conditions, this can lead, for example, to some initiation of epileptic type activity. The two populations that I'm describing here, and Paul in his talk describe also the effect of gap junction, which are coupling not the neurons but the glial cells around, are very important actually for the regulation of these ions. Okay, now let's go to the microscope. Yeah, this is important now. Let's go to the description of what happened at a molecular level. Well, if you want to describe, and this has been done quite a lot with one of the best group looking at the questions in Chicago and around the Midwest about how an ion goes through the channel, what is the right description. And so I'm interested in here about what happened after, when they are going to be located in the dendritic spines. So the classical model is you can use a small Hofsky limit of the Langevin equation, which is friction, potential, plus the random noise. Now the question is, of course, what is the electric field? What is going to, how is it generated? Obviously, you need to account for all the ions and possibly water. So this is something that looks quite complicated. So this is a question I don't know how to answer, but let me ask the question. How do we need water molecule to be added in the model? Probably yes. How to do it? I have no idea. Probably there is also some local polarization of the medium. You can think about dipoles, especially when a current enters, that probably will affect the distribution of ions. This is not clear how to add it here. And finally, if you think about electrolyte, and again, there's a huge literature about electrolyte, especially for battery. Batteries are very important. There's money involved, and how to design batteries that are efficient is a great subject, especially of modeling, to understand how you can reduce the resistance in a clever manner. So, yet it has been shown a couple of years ago that you can get gradients of concentration. That is, there is the species are not uniformly distributed, and it can affect synaptic transmission. This is a group of Dmitry Ruzakov at UCL who started to investigate this both theoretically and experimentally. And now I would like to discuss, let's start with the failure of the electron neutrality assumption and see what has a consequence for what we can predict about the organization of the ions. Right, now I'm going to start the talk. This was just the background before. Plan. So let's discuss a model of non-electron neutrality in confined microdomains. Then I would like to discuss Poisson, and PNP means Poisson Nernst Planck with small hole and the escape of an ion that is driven by the field. And then I would like to connect to new data and explain how the data itself generates a kernel for deconvolution in the spirit of this data, large data analysis, where you let the data define for you the kernel that you're going to use. Then I'll explain in details to extract information about the electrical field in nanodomains, in microdomains. And finally, we will show some simulation about this PNP in the shape of a dendritic spine. The basic idea is that the classical cable equation, which was true for cable, do not apply anymore for a dendritic spine because there is a shape and because there is a small hole. Now, how do you address the question of the geometry? Without the cable equation being valid, you cannot use any more Hodgkin-Huxley. So then that's why you have to go to PNP, Poisson, and Planck, where you look at this description at this intermediate scale, not at stochastic level and not at the bulk level. Okay. So let's look at a simple question. If you have a box, a ball, and you throw diffusing particle, you wait steady state, you expect the sum times, which is given by the first eigenvalue of the Laplace operator, you get uniform distribution. This is steady state. Now what happens if you have a field? So suppose you have an electric field and one type of charge, only plus. So then you have to rewrite the equation. Instead of having the diffusion, this is classical PNP. The Fokker-Planck equation has diffusion plus drift, and so this is the zero flux. No ions can go out, and you have total conservation of the particle. And now the problem is the field is satisfy Laplace equation, the Poisson equation, which connect the density to the field. And then you have the boundary condition, which is dn equal minus sigma, and the sigma is the total charge divided by the surface. Now at steady state, this is 0, and you can, in this domain where you don't have holes here, you can integrate. This will give you the classical Boltzmann exponential law, which is written here. This is rho equals this at steady state. And now if you want to understand what is the field, then you come up with a very nonlinear partial differential equation, which is Laplacian phi, equals exponential minus phi. And here you see it's a non-local term. Here you have to integrate over phi. And now you are left with solving this equation to have the distribution, to have the distribution of the field first, and then this will give us the distribution of charges. Let's analyze this equation in dimension, in the case of a ball, with radial symmetry. So the Laplacian reduces to this term. And now you are left with the Neumann boundary condition at 0 because of symmetry. And then this is the compatibility condition, d phi dn equals this term here at the boundary. What are the solutions? Well, first of all, this is a non-trivial problem to solve numerically in any dimension. If you look at this, this is an overdetermined problem. You can fix the potential because it's defined up to a constant. So you have u0 equals 0, u prime 0 equals 0, and the boundary condition. So you have 3 constraints, and it's a second order differential operator. You can, so if you think about even doing it numerically, except in dimension 1, where this drop, and you can just integrate by multiply by u prime. For dimension 2 and 3, you need tricks. Because again, if you have the solution here, you have the integral of the solution in your entire domain. I'll reveal the trick once the paper will be accepted, because it has been rejected in Siam Journal of Applied Math, because the reviewer could not understand the difference between gravitation and electrostatics. This is first year university classes in physics. The problem is that the editor didn't say anything.
It wasn't me, David.
I know. Paul, I don't blame you for that. I know that it's not you. Yeah. Okay. Dimension 1, this is a solution. And you see it's log of cosine of lambda, there is something here, lambda divided by I lambda, where I lambda is this integral here, and it satisfies, because of the boundary condition, a transcendental equation. And basically, you can see that lambda is the total number of charges. When charges goes to infinity, basically what you see an accumulation of charges at the boundary. And this term cannot go above pi over 2. So this is not, you don't get cosine being equal to 0, because then you get the log, and that's why you get the singularity. But now we understand that it goes like with the log. This is asymptotic expansion of what happened when you reach to the boundary. Of course, if you go beyond pi over 2, you get in singularity. This is an imaginary solution. This is for us. but this is a physical one at the limit. Dimension 2, it's not very hard because you can reduce to dimension 1, and again you see log with this behavior. X now is normalized. This is the radius, normalized radius of the ball. Now in dimension 3, this is also in dimension 2 actually. This is really a curvature. This is known in Riemannian geometry. This is really like a curvature problem with a minus sign. And this is also equation that were known in reaction diffusion for flame propagation. And we used here what is called the Gelfand coordinate that he introduced a couple of years ago to transform this equation into a dynamical system. And the idea is that is this behavior in the log that you see here true in dimension 3, right? Because we're interested in dimension 3. So then for this, you change really the aspect of how you look at the problem. It now becomes a question in dynamical system. And so basically, we are in this quadrant here. And if you start with any point here, actually the only solution that matters, because you know the boundary condition, which is u0 equals 0 and u prime 0 equals 0, this is equivalent here to this to the center. So the only possibility that you will reach to this point, starting here, is to be exactly located on the separatrix. This allows to say that there is only one solution, contrary to what happened in the opposite cases. When you start in here, in the case of the flame, if you draw a line here and you count how many times you are going to hit or to cut this red separatrix here, you see you can have 2 points, so you can have 2 solutions. This is not the case here. And so we have an asymptotic argument. We have no proof, an asymptotic argument that in dimension, this should be 3 here, u3 of x goes again like log, and the function f of lambda that we could calculate in dimension 1 and dimension 2, we don't know how to calculate in dimension 3, but this is the behavior. And again, this is an argument in the paper so that we have this, this behavior. So dimension 1, basically you have a field, and you can see the effect. So this is, we can calculate the limit of this coefficient mu. You can see the effect of the distribution of the field. This is for high lambda, for high quantity of charges. Dimension 1, dimension 2, dimension 3, just this term that the limit mu lambda is changing, and you see here an accumulation of charges. Now, if you remember your classical electrostatic, what is the, how do you characterize capacitance? This is voltage proportional to a constant, Q, I forgot no mine. Total charges equals C multiplied by U, right? So there is a linear dependency between the total number of charges and the voltage. So U0 minus U1, this is the voltage at the center of the ball, and U1 is the voltage at the periphery, so a point at the periphery. Dimension 1, dimension 2, dimension 3, and you start to see here the effect of putting a lot of charges, which is the effect of saturation. And this is the dimension 3 cases where you see that the numerics and this ansatz is working very well. Okay, let's summarize the idea here. Diffusion, everything is uniform. P shows for the case of a ball with no absorbing and no fluxes at the boundary, that first of all, the difference of potential between the center and here behave like log of this n-dimensional everything. R is the radius, Q is the total number of charges, T temperature, Boltzmann, E electron charges, maybe Q is N here, this is the total number of ions, epsilon permittivity, epsilon 0. So the voltage here is a function of the total number of charges. You see that if there are little charges, they behave like diffusion. If you have a lot, which we can quantify here, you can see this effect. And this is significant because look at this. If you think about neurons, about tens of millivolts, you can go from 0 to 40 millivolts with this amount of charges, right? And this is a deviation that we see from here, from the classical electrostatic theory, where this is proportional, with where Q and voltage are proportional, and here we see the deviation. All right, so now we have this as an intuition to start looking at real data. And okay, before doing this, let's discuss now what happened. If you're interested, finally, biology is by definition transient. Everything is moving all the time. We don't have steady state. So now let's suppose you send a current, you have, and you look at the distribution of charges, and the question is, what defines the voltage when now we have a possible flux? So in particular, let's look at how long it takes for a particle to escape here through a small hole, which is going to be the beginning of the spine neck. So now we have to solve the why calculating the time, because this will give us the time scale of electrodiffusion, not diffusion, electrodiffusion. So you solve the classical mean first passage time equation with the field and with reflecting no fluxes everywhere except here at the beginning where we have the dendritic spine. Now what happened? If you have Brownian diffusion, it will the particle will basically fill the space before exit. Here, because of the effect of the field, when you start very close to the membrane, you stay on the membrane, right? Because what happens if you start in the middle? In the middle, the field is going to zero. But when you are confined, because the field is very strong, you are going to stay very confined. And then the particle is going to move along the boundary. I mean, the ions will move along the boundary and then escape. So this is the idea now of seeing this by doing some asymptotic when you have a large field. On this equation, what's the mean first passage time is doing? So basically, let's unfold the ball and put everything on the plan. You have the dependency in the z direction and the dependency in x and y direction. And to here make a long story short, basically think about this as the second derivative in z, where you have the field only in the z direction. And then you have the two-dimensional Laplacian. Now, when this is very large, you can show that the asymptotics is more or less of the time is exactly the time for a particle that would have moved only on the surface because it is confined. And this is good in first approximation, so you just have to solve basically the mean first passage time equation on the ball with a small hole, and this is known. We and others have calculated it. You just get the mean first passage time in 2 dimensions to a ball. Suppose you start at an angle theta, and the characterization of the hole is the angle delta. And so this is the mean first passage time that you get. It's log of 1 over delta. So dimension 3, mean first passage time goes 1 over epsilon, 1 over delta. In dimension 2, it's log. So you have reduced quite a lot the escape time. Now it's very fast, which makes sense because we know that electrodiffusion is much faster than pure diffusion. Here you reduce because you see it as a reduction from dimension 3 to dimension 2. And now you can use it if you have n particle, independent particle. This is the time for the first one to escape. And now you can calculate the flux, which is 1 over this time. And now you get the flux and see that how it depends on the geometry. So let's conclude this part. The amount of charges inside define the difference of voltages and the distribution of charges. The current here is defined by the fact that we have a small hole. So it's 2 different geometrical structures. One defines the voltage and the other defines the current. And still, you know, we are interested in the relationship between current and voltage. This is what electricity has been all about. If you have a resistance, if you have a capacitance, and if you have all kinds of impedance, you'd like to connect the voltage to the current. Okay. Now we have these basic concepts in mind, and now let's explore data. And the idea is how are we going to extract the electric field in a structure where you cannot access directly. Because this is a neuron, and this is a pipette. So you're sticking this small electrode here, this small pipette. This is in the soma. While we are interested in structure like this, you cannot touch a small dendritic spine. So you don't have direct access to the electricity here. So how do you do this? Well, there was a revolution in the past couple of years. There is a lot of revolution today, right? One today, one in so this was the revolution of arc lights. There is a class of dye that are sensitive to voltage. That is, they emit light when there is a change in the voltage between outside and inside. And you can see this is an example. If you go from minus 100 millivolt to 0 millivolt, so you have a step function, and you see how the dye responds to it. So it is quite slow. Remember, I ask you to pay attention to the time scale of electricity, of an action potential. It's 10 milliseconds. 100 milliseconds, this is already something big. But few milliseconds. Here, we're talking about half a second. All right, so this is one step in one direction. If you do a step in another direction, and you can see it is still the response of this fantastic reporter is much slower than the response of the electrical signal that we're trying to extract the information from. Okay, so then we're interested. So this is an example of a dendritic spine. This is a dendrite. This is where the pipette is recording. So this is a recording from the soma here. And this is what happened in this small structure. So this is blue is the spine, and red here, this is the change in the intensity of the reporter in the dendrite. Now, how can we connect? So this is there is no information here about voltage that has to be converted. This is just fluorescence. So this has been solved. And there is no also information about time, because I just show you that the reporter is too slow. So the idea is that, is there a way to use the convolution or deconvolution, or let's say the connection between the signal here and the signal in the soma that you get from the classical recording? In other words, if we would like to have information about something that we cannot record, we are not sure about the time scale, we're not sure about the intensity, how can we get something where we can calibrate this information to create a deconvolution kernel that we can apply here? So the idea was the following. Just use the soma where you can stick an electrode, and now we can compare two signals. The signals from the electrode and the signal from the fluorescence intensity. And now the nature has done for us the deconvolution. The deconvolution is to go from that signal to that signal, and that's it. So now you can, and I'm going to describe how we have deconvolved this signal. And once we have understood how to do this in the big domain, we're going to apply it to the small domain, and now extract electrical activity in a dendritic spine, in this small structure. All right, so this is the signal that you have. So Tom had the beautiful methods at the same time to approximate and to get the parameters. Here what we did is we use a classical approach. First, we use a special filter to get rid of, really to smooth out all of these fluctuations. So this is the signal that we have in the dendritic spine head. This is the signal in the dendrite. And then what was important is to find an analytical basis to parameterize those responses. So we came out with one. I'm sure there are many. And then, so this is now the signal. And from this signal, which is too long, we would like to go back to the real time scale. Now we are going to use the kernel that comes from the data. The data is doing, I mean, the comparison of the two experiments is giving us this kernel. And now what we just need to do is very classical deconvolution, is to find if we know J, we have to find here this function f. And so we use the kernel, and we basically could calculate everything. So then we can go from the fitted signal, which is what you have here. First of all, we now convert into voltage. And now if you see the scale here, this is now the final result of the deconvolved signal. The blue has been moved. Everything is now much faster, as you can see from here. And this is now the deconvolved signal, which is now the voltage, the difference of voltage between the spine head and the dendrite. And to our surprise, you could see that it is significant. It's from 5, let's say, for example, to 25 millivolts. And with a time scale of hundreds of, much less of than hundreds of milliseconds for the peak. So this is obtained by a protocol where you induce here, you release glutamate. Glutamate is a neurotransmitter that will open here the receptors, and then you will have the flow of the current, and the reporter is reporting for us this flow of the current, this change of the voltage.
Can I ask, where did, I mean, 25 is much bigger than the 4 and the 1 that are measured. Maybe I'm just missing something.
No, this is df. This is the signal intensity. This is df over L. So this I haven't discussed. I'm sorry. Yeah, there is a way to convert. There is a way to convert. I'm not presenting it here.
Because even on the rightmost picture, if you look at the broken green and broken red,
yes,
they are also very low.
Yes. Yeah, this is. Yeah,
okay.
let's yeah, this I'm not going to discuss. This was the two things I wanted you to pay attention on. All right. So again, yeah, it's a good point. The conversion here, this is a separate story. All right. So now we go back to a small structure. We have the information for this region of interest, and we can basically do the deconvolution. We have this low signal that has been deconvolved or into this green one, and this is for the soma. And now we do the same for this dendritic spine. So you had from the head, you go from this data to the deconvolved one, and now the same for the dendrite. You go from this data that you acquired, and this is the result of the deconvolution. And this is another example of a different dendritic spine, where you see here now the voltage is not any more than of the order of 20-25. It's much reduced. Yeah?
Sorry, do you stimulate only one spine at a time? Or
Yeah, no. This is, I'm going to show several now.
Okay.
This is one spine, and then we are going to do that for several of them, of course.
Okay, so the convolution changes. Then.
Yeah,
Okay.
each time you have to do, the kernel stays, because the kernel is giving you the, this is from the soma, so it gives you the time scale and the process, and of course, we assume that what happened in the soma in terms of the distribution of dye is the same as in the dendrite and in the spine. Yeah, I'm missing a slide. Okay, never mind. Okay, so now can this be explained, this difference, by the classical theory? Now we go to cable theory, and we're going to use, I mean, I'm not going to present you the results of the cable theory, but basically we wanted to, I'll go directly to PNP. Can this drop here be explained by the classical equation of electrodiffusion? So for this, we had to incorporate in the Poisson-Nernst-Planck. We had before only what the positive charges. Now we added the negative charges, and the idea was to look at what happened inside the neck that connects basically the head here to the rest of the dendrite. So the idea is that now you start with some you have this initial condition. You have the boundary condition that you can impose here for the voltage at the head for the different concentration. You assume that there is a current flow at the head, and again you have to impose some fixed concentration here at L for the positive and the negative charges, and V, the potential, is defined up to a constant, so you can always say that it is 0 here. And now what we have to do is to solve the Poisson-Nernst-Planck equation for this structure, and now the electrical current is going to come from the data. So this is the empirical electrical current, and if we solve this equation, we should be able to predict or to calculate the voltage and see if this is sufficient to explain what you see from the experiments. All right, so this is I recapitulated here the frame. This is the head, the neck, the dendrite. It's approximated as a wire. We neglected here at this stage the effect of the head, and now you want to compare the difference of voltage that you have measured experimentally, which is the red curve, with the computed one. And so from this calculation, from the solution of this PDE, we could then extract information about what is called the capacitance and the resistance of this structure. And in addition, not only we could look at the voltage, but also now you can calculate the current that is really that is flowing inside the spine neck. So now this is the answer the answer to the question that you have asked. Then we repeated this for different spines, and of course there is a large variability. So again, to look at that precisely, we looked at there was this deconvolved data in blue, and this is in red the result from PNP, and you can see when it fails a bit and how it works. We looked also at the classical electrical properties like circuit approximation. We looked at the cable equations, and it didn't work. I didn't show you the result here, and I will show you why they are not working. This is due to the fact that you really have no constant concentration. You have really some gradient, especially when you have a flux of current coming from the spine head.
Can I ask?
Yes.
I mean, as far as data simulation goes, you obviously clear that it's working, but you also need to kind of have initial conditions when you start, and they should not matter, or you must be very good.
Yeah, okay. All right, I have to explain more about how we did all of these. The initial conditions are coming. Okay, all right. Let me spend 1 minute on this.
We can talk about it at the end.
Let's spend 1 minute. So how you get the initial condition? You look at the fluorescence in the head. The fluorescence in the head will be the initial condition for what happened in the spine neck. So this is how you get the initial condition, and then you can inject this into the equation. Yeah, this is an important point. Yeah, okay. We have seen this, right? Yes. Okay, and so now let's go into some more statistical analysis. How long do I have? 3 minutes. Okay. So this is going to be probably the last slide. You can estimate the voltage in the ball using here simulation. This is PNP simulation. I'll maybe describe a little bit of this. But what is the most important in all of this is to look at the difference of concentration of the positive charge between what you have at point L and at point 0 as a function of time. You see that it's significant. Out of, let's say the basal level is about 160 millimolar, and you can see here a difference while this occurring of 30 millimolar, which is significant. It's not just 0. So there is a difference in the gradient, in the concentration, the gradient concentration. There is a gradient concentration here inside this little nanometer dendritic spine. And so one more thing is that the IV relation, you remember I showed you with this very naive preliminary calculation for a ball about this possible nonlinearity between V here and charges. It was charges. Now it's now voltage. It's now current, and you still get here this very nice nonlinear behavior because the voltage-current relation can be approximated as constant, and so you can extract the resistance only for some the tangent when you are very close here to 0. Okay, I'll skip the rest. Now you can now do full simulation of P and P, and the output of all of this is that if you inject a current here in the head, the voltage drop in the head is almost 0 here inside. It's almost 0, so there's no resistance from a ball. Most of the resistance, most of the drop that you can get will come from the length of the spine. And from this, you can do actually a reverse engineering problem. If you know, for example, the difference of voltage, you can measure, you can estimate what was the initial current that was injected inside the spine. Okay. Again, I'm going to stop here. I would like to thank all of you. Particularly, I would say Radek, Costa, and Sam. Radek and Costa had the idea of the organization of the program, and they did a lot because they are living in the UK. It's not an excuse that we did much less, but they did really a lot. Of course, Sam organizing quite a lot. I thank really my collaborator for this. And as I told you, I just take one more minute. It's not part of my talk now. It's part about spreading some ideas. I was in contact with the editor of Cell. I tried to push forward of a new journal saying that this community, people that do modeling, simulations, data analysis, cannot reveal new information from biological data. So there is, we think, there is a room for high-impact a journal where we could publish a method, simulations, showing that this simulation of this method was used to solve, and this is should be figure 1 of the paper, an important biological question. So it's still under debate if we continue with Cell or if we go to Nature Press. I know that our friend from Columbia and from Yale, Jim, who has the Nobel Prize, can fight for this cause. But we need more people of this, with this vision to help us to push, because I think it will help this entire community to be visible. So again, I will call for associated editors. I'll be happy if you'll be interested. And I'll be happy also to have more help, because we probably need to have a very senior person that can put the case, show that it is important. And on this, thanks for all of you for coming, for participating to this week, and for the entire program. Thank you.
Any questions for David? Okay. Well, thank you all again for coming. I hope you enjoyed the workshop. I guess have safe travels home. And yeah, and to all the program participants, thank you all for coming to the program.